But let's just move to a real example and see how well you can do with the analysis. So there's the first 4 differentials of this particular set. And now you have to decide whether that's high or low, high or low, high or low, and then come up with a pattern and write that down. Okay, I move on. You all happy with the first one? Second set is R11, 6, 8A, and 9G. I think that's fairly easy. Everyone got that? Third set, SR36, 9B, 30, and 17. The fourth one, interesting gene this, SRTMP, very topical lately. Especially in the African context. And the last set, SR24, 31, this is the gene that created all the interest in UG99, TTKSK. SR38 typically gives a kind of an X reaction when it's low. It can also give a flag to certain pathotypes. And this is our susceptible control, McNair. It's got a gene, but I don't know of many isolates that are actually avirulent on McNair. It's supposed to be a susceptible check. You probably can't see that very well. So now you wrote down all the patterns. Now you have to allocate, according to your lows and highs, you have to allocate a letter and come up with a 5-character race code. You all got it? So pathotype 1, high, low, high, high. You all had that right, I suppose. Did you? Robert did. The difficult one was the second gene, I think, SR21. It was intermediate, but still low in this situation. High, 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 low. Goes out to S. Low, high, 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 K. S, C. So the race code will be PSKSC. And this is a real South African race, quite different from what we hear. We only hear about the T races and the R races. But this is one of our old ones, not important anymore, but...
They called SR17 lows, they can set through.
You did?
So I had low, high, high loads. So I call it PSJs. Which is actually what it makes you.
Yeah. You can see the islands?
Really?
Yeah.
So you, SR17 you called high?
Call it low.
You called it low. You're right, that's what I called it.
Yeah, exactly.
I call it high, yeah.
It can be a tricky one. It's a mesothelic gene, and it can go pretty high.
And the reason why I call it high is because I know what the low looks like under my condition.
Yeah.
And it's different from that. Okay, let's go to another example. Different race. Maybe you want to go back to the first one now that you've seen the susceptible. Set 3. There's an interesting one. See, there's 17, Robert.
Yep.
Mm-hmm. Set 4. I think that's again straightforward. And the 5th set, also quite easy. So in this case, you can see SR38, and the differential I used was Trident, Australian variety. Trident also has another gene or genes, and it gives a fleck on Trident, not the typical X. Okay, we need to first go back to the code again. So you can key out the race. Okay. Yeah. It's not really a difficult race, I think. Fairly avirulent. You had a B. Who did not have a B? All B's, huh? High, low, high, high. P. That was fairly easy, I think. G. S. And a C. That was low on SR30, but that's a difficult gene for me to score. And again, you've got to repeat until you're fairly sure. And you've got to do side-by-side comparisons with isolates you know that are high on the gene and ones that you suspect are low. That's the only way you can come up with a definitive answer. So it's not a first-time thing. You've got to repeat. Yes? Second set. High, low, high, high.
The first one, third set. Second set. The first one was supposed to be somewhere in between.
Yeah, but again, my pictures are not always perfect. And in this particular one, the light, the natural light that came in, I think washed out the color on the first leaf. It was closer to the window. It should be the same as that. SR11 is susceptible there. So there's the difficult one. I know that this race usually gives a low reaction on SR30. That's why that should be low. But if you had it high, it's probably okay in terms of what you're looking at just on the slide. And again, this is also a real South African race. We have 3, possibly 4 pathotypes within this race cluster. They are actually all virulent on the triticales. They have virulence for SR27, they have virulence for the gene in kivit, which we don't know what the name is. It's virulent for SATU. And it's avirulent on 5, so it's quite different from the others that we have. First set. That's a low for 7B. It could also be a difficult gene to read. And a lot depend on your inoculum concentration, how many spores you put on, and how well the infection types developed. Okay, easy one. Pathotype 3. And have a look at this one. Controversial gene, SR21. Second set, easy. Not going to waste time on that. Third set, easy. Low, high, high, high. Fourth set, also easy to read. And you can already see, I think, a lot of the genes are ineffective to this particular isolate. You can see all the highs coming up. (24, 31, 38, and McNair, your check. So when you have all of them high, it's a T, okay? All right, you all got it? What's happening now? Right? Yeah, all at a T, I'm sure. Also a T, K, S. Oh. You thought SR31 was susceptible because you're so programmed to think of TTKSK. This is the first UG99 race we detected in South Africa in 2000. It's avirulent on 31. But it belongs within the race group. That's been shown over and over and over with the molecular markers. And
so it's the molecular marker that defines the race group and not a set of differentials.
absolutely. And I think that point was also made previously. I think Sajid made it. It could have the same race number, but genetically they're actually quite different.
So there's a cluster analysis, and how does that work?
Sometimes you have very clear allele pattern.
Yeah.
You don't even need to do some analysis. So the first thing is if you have the same multilocus genotype. So means like if you have 20 loci, they're diagonal, you compare the alleles, they have the same allele patterns. So that's the easiest way. And with Warrior, we know the multilocus genotype we run in the lab and we say it's Warrior type. But then if you have some variation, then you go for analysis. And cluster analysis is one of them, but there are so many.
And the cluster analysis gives you the dendrogram.
Yeah. And but you can also use other analysis, and they can exactly tell how distant they are. Or you can use some even ancestral analysis. They can tell you that how far in generation they're related.
So you really can't declare it to be in the UG99 race group until you have a whole genome.
Yes. And this is why it's an F. You can see that's low, low, high, high. It's not a K, then it would have been low, high, high, high.
But just coming to your question, it would be interesting to see if this virulence to 31 is present in some other genetic group, and Les can tell us because he has this worldwide collection.
Well, they've not detected SR31 virulence before.
Yeah, but means even now.
Only once, Julio reported it once before, but the culture wasn't retained, so we would not report it.
Yeah, but no, at this time means in the current population you can still have subgroupings, and you can see the virulence is, so it could be a recombination between the UG99, but you can have that virulence in another genetic group.
There's no evidence of that having occurred with the UG90, with the SR31 virulence, I think.
Yeah.
The comment I was going to make is, so you'd use the markers, and based on percentage similarity, right, so you can have the same multilocus genotype, which means basically two isolates are 100% the same on their molecular fingerprint. So we would say that they're part of the same home lineage. They may differ in their virulence, but their genetic background is 100% the same. But equally, I could go back to Australia and do a race analysis on a sample and find a pathotype TTKSF, right? With the same virulence as this one, a completely different molecular genotype, but the threat to farmers would be the same. I mean, it's a different genotype, molecular genotype, but it has the same virulence. So in reality, in the field, it poses the same threat to growers as the one that's active.
Right? Slightly different presentation. I hope you can see set 1. Now I'll put them up all up for you. Interesting one there. This is also a real-life example. All of these actually are. So I think the obvious lows would be that one and these two. The others are either susceptible or inconclusive, high intermediates. Okay? You all had a T, I suppose? Another T? Okay. You all had a T, huh? I also had a T, because I know what SRTMP should look like. And then F, low, low, high, high. And this isolate came from Zimbabwe a few years ago. And we started the conversation, the emails, is this high or low? Is SRTMP environment sensitive? Is it expressing well? And I still haven't figured it out. But it could be potentially different from the others that we have based on the SRTMP, the triumph gene response. Which one you want to go back to? 4. Oh yeah, we only have the I should actually go back to the previous race so you can see what that gene looks like when it's really low. So you remember that response? It's a high intermediate.
Yeah, it's like intermediate.
When you go back, it usually gives us a low 2, even a 1 when it's low. So that's why I call it high in that experiment, compared to what the gene should look like. All right, so that's StemRAS. Any questions on the StemRAS system? I think you've all got it, and it depends on the quality of your infection. The quality of your infection depends on the quality of the seedlings you grow, the purity of your isolates, and your RAS methodology and experience.